Hello everyone. This session gives you a broad overview of the main in vitro model systems used to study the gut-brain axis, starting from the simplest 2D cell line approaches right through to cutting-edge organoid and assembloid platforms. And by the end of this overview, you should have a clear mental map of the model landscape, understand the core trade-offs between complexity and practicality, and feel confident about which approaches are most appropriate for different research questions. So before we look at the models, let's just make sure we're on the same page about what the gut-brain axis actually is. At its core, it's a bidirectional communication network linking the enteric nervous system, sometimes called the second brain, with the central nervous system. The signals that travel along this axis are diverse: neural signals via the vagus nerve, endocrine signals from enteroendocrine cells, immune mediators, and metabolic byproducts from the gut microbiota. Key molecular players include serotonin, of which around 90% is actually produced in the gut, as well as GABA and a range of cytokines. And why does this matter clinically? Well, dysfunction of the gut-brain axis has been implicated in conditions as varied as inflammatory bowel disease, irritable bowel syndrome, anxiety, depression, and cognitive disorders. And developing good models to study it is genuinely important for both basic science and drug discovery. And this slide gives you the big picture. We have 5 major categories of models arranged here from the simplest on the left to the most complex on the right. So you have 2D cell lines, cell cultures, 3D spheroids, organoids, and assembloids or organ-on-a-chip systems. The table at the bottom captures the key trade-offs. As you move left to right, physiological relevance increases, but so does cost, and throughput drops sharply. There is no universally best model. The right choice depends entirely on your research question, your budget, and the stage of your project. And we'll now walk through each of the category in turn. So 2D cell line models have been the standard of gut-brain axis research for decades. The most commonly used lines are CACO2, which model intestinal epithelial barrier function, SUSHI, a human neuroblastoma line used as a proxy for dopaminergic neurons, and STC1, which are widely used to study enteroendocrine cell signaling. These models are best used for mechanistic pathway studies, initial compound screening, and toxicity work, where speed and scale matter more than the perfect physiological mimicry. And the advantages are hard to argue with for early stage work. These cells are cheap, well characterized, easy to genetically manipulate, and scale up beautifully for high throughput screening campaigns. The limitations, though, are significant. Cells grow as a flat monolayer with none of the architecture of real tissue, and there's no communication between gut and neural compartments in a single dish. And after many passages, CACO2 in particular can drift phenotypically. So a step up in complexity. Cell culture and transwell systems let us put multiple cell types together, typically an epithelial layer on a porous membrane with neuronal or immune cells in the compartment below or mixed together. This configuration opens up some genuinely interesting biology. You can measure barrier permeability in real time, look at how inflammatory mediators from immune cells modulate neuronal activity, and model how enteroendocrine cells communicate with nerve endings across a physical barrier. That said, these systems still sit in 2 dimensions, and maintaining the viability of multiple cell types over extended periods is technically demanding. Standardization between labs is also a known problem. Protocol differences make it hard to compare results across publications. And the best use cases here include barrier function studies, neuroinflammation experiments, and mapping the signaling crosstalk between the gut epithelium, immune cells, and neurons. Moving into 3 dimensions, spheroids are self-assembling aggregates or cells grown in suspension or embedded in extracellular matrix such as matrigel. Enteroids are specifically derived from the intestinal crypts and retain some of the in vivo-like crypt villus organization. The jump to 3D brings real benefits: more physiologically relevant cell matrix interactions, better recapitulation of oxygen and nutrient gradients, and improved predictive power for in vivo drug responses. They're also compatible with live confocal imaging, which is useful for tracking dynamic processes. However, spheroids are not without frustrations. Size and composition can be variable between experiments. Large spheroids often develop a necrotic core due to limited nutrient diffusion, and accessing the lumen of an enteroid, which is sealed off from the outside, can be technically tricky. These are particularly good models for studying drug absorption, crypt renewal dynamics, and early neural differentiation. Organoids are arguably the most transformative tool to have entered this field in the last decade. They are derived either from plant tissue biopsies or from induced pluripotent stem cells, and they self-organize into structures that closely resemble their tissue of origin. For the gut-brain axis, we have three key types. Intestinal and colonic organoids form villus crypt structures with LGR5 positive stem cells, a functional mucus layer, and a measurable barrier. Brain or cerebral organoids develop cortical layers, distinct neuronal subtypes, and region-specific identities, though they remain somewhat immature. And enteric nervous system organoids contain functional neuronal and glial networks capable of generating peristaltic-like contractions and releasing neurotransmitters. The headline advantage is physiological fidelity. These are the closest we can get to real tissue without using an animal. They also enable truly personalized research. You can take a biopsy from a patient with Crohn's disease or Parkinson's and model their specific disease biology in a dish. The trade-offs are real, though. Organoids are expensive and technically demanding. Batch-to-batch variability can be a headache. They lack integrated vasculature and a full immune complement. And for cerebral organoids in particular, there are genuine ethical discussions ongoing about the moral status of these models as they become more sophisticated. At the frontier of the field, we have two next-generation approaches. Assembloids are created by physically fusing gut and brain organoids together and allowing them to integrate. The resulting structure captures bidirectional signaling between the enteric nervous system and the central nervous system compartments in a single model. Researchers have used assembloids to study how enteric neurons project into and communicate with brain tissue, and to model disorders like Parkinson's disease and autism spectrum disorder, where both gut and brain pathology coexist. Gut-on-a-chip and microphysiological systems take on a different approach, using microfluidic devices to apply physiologically relevant shear stress and peristaltic motion to the gut epithelium, and to co-culture it with microbiome, immune, and neuronal compartments. Some groups are now linking gut chips to brain chips via a shared fluid circuit. The caveats, however, are significant. They cost very highly, there's a steep technical learning curve, and reproducibility challenges. Also, regulatory frameworks for using this data in drug development are still catching up, but the trajectory is clear. These are where the field is heading. So how do you choose? The summary table is meant to be a practical decision aid. If you need speed and scale, early compound screening, target validation, then cell lines remain your best friend. If you need to understand barrier function or two-cell type crosstalk, then co-culture systems are the next logical step. 3D spheroids add extracellular matrix complexity for a modest cost increase. And organoids are the go-to when patient relevance or disease fidelity is paramount. Assembloids or organ-on-a-chip systems are for translational questions where you genuinely need the full gut-brain circuit. One key message to take away here that these models are not mutually exclusive. Best practice increasingly involves simpler models first to generate and filter hypotheses, then validating findings in progressively more complex systems, and that a tiered approach makes your science stronger and your budget go further. And I want to end briefly by looking at where the field is heading towards. AI and machine learning are beginning to transform how we analyze organoid imaging and multi-omics data, helping us decode the complex signaling patterns these systems generate. Vascularized organoids incorporating endothelial cells and a blood-brain barrier component are an active area of development that could address one of the biggest current limitations. Large-scale patient iPSC biobanks will enable population-scale gut-brain modeling for the first time. And on the practical side, standardization is a pressing priority. The field needs consensus protocols and quality control so that data from different labs can be meaningfully compared. And regulatory agencies like the FDA and EMA are actively developing frameworks to accept organoid-based data as evidence in drug approval processes, which will be a major driver of adoption. Finally, truly physiological microbiome integration, long-term co-culture of live, diverse microbial communities with host organoids remain an open challenge, but one that several groups are making real progress on. And that brings us to the end of the overview. I hope you found that useful and it gives you a foundation for thinking about which model systems might be most relevant to your own work. Thank you very much for listening.